Thank you very much. Thank you to ARM for hosting today. It's a great event. I've been here for about 6 years now, and it's so exciting to see all the progress. As she mentioned, my name is AJ Bergmann. I'm the Vice President of Finance for Capricor Therapeutics. My CEO, Dr. Linda Marban, is currently in France at the World Muscle Society, so she was sent there, and I was sent here. So it's my pleasure to be here today to tell you a little bit more about Capricor. We're a Los Angeles-based biotechnology company currently focused on the treatment of rare pediatric disorders. We are a publicly traded company. We're listed on the NASDAQ. Our ticker symbol is CAPR, so I will be making forward-looking statements today. So a summary of Capricor. Capricor was founded in 2005 out of Johns Hopkins University. Our founder, Dr. Eduardo Marban, was head of cardiology at JHU and now is the director of the Heart Institute at the Cedars-Sinai Medical Center. That's where Capricor is located. That's where our lab and manufacturing facility and corporate offices are housed. We have a pipeline focused on rare pediatric disorders, which I'm going to talk about in a few moments. The lead program is in Duchenne muscular dystrophy. We've demonstrated clinical proof of concept in a phase 1, phase 2 study. We announced the 6-month full results this morning at the World Muscle Society International Congress meeting in France. We have potentially a registration study starting in the first quarter of 2018. It's obviously a large opportunity with the favorable pricing scenarios in orphan drugs. We have a scalable, cost-efficient manufacturing process and development and significant ownership by insiders and a strategic. So Capricor has 2 products in our pipeline. I'm going to spend the majority of the next few minutes talking about our lead program, CAP1002, which are allogeneic CDCs or cardiosphere-derived cells. They're in clinical development for the treatment of Duchenne muscular dystrophy. And as I mentioned, we recently completed 6-month results in our first phase 1, phase 2 study. Our second product in our pipeline are what we call CAP2003. These are exosomes. Our exosomes are derived from our cells. Exosomes are a nano-sized lipid bilayer vesicles, and we believe to be the cell-to-cell communicators. We are exploring a variety of indications with our exosome platform technology, looking at inflammatory disorders as well as an ultra-orphan disorder known as hypoplastic left heart syndrome. We have approximately $4 million from the NIH to conduct preclinical and clinical work, and we hope to be advancing that program over the next few years. So what is Capricor's technology, CAP1002? As I mentioned, it's an allogeneic cell therapy. It's a unique population of cells, which includes cardiac progenitor cells with a distinct surface marker profile. It's been the subject of over 100 peer-reviewed publications. They were discovered by Dr. Eduardo Marban at Johns Hopkins through a collaboration with the University of Rome. And they've been evaluated in several clinical trials to date, post-myocardial infarction, heart failure, Duchenne muscular dystrophy. So we've been in 4 clinical trials to date. And they show, a variety of different things, of course, predominantly being regenerative, anti-apoptotic, anti-inflammatory, and anti-fibrotic. And we believe that the cells go in and release exosomes, which secrete a variety of signaling molecules. They do not engraft. So a little bit more about Duchenne muscular dystrophy and Capricor's opportunity. So for those in the room that aren't aware, I think most people are, what Duchenne muscular dystrophy is. It's pretty front and center in the Wall Street arena these days. It's a rare pediatric disorder. It afflicts about 1 in 3,600 male births. There's approximately 15 to 20,000 boys in the United States with this disease. It's an X-linked genetic deficiency where there is a deficiency in the dystrophin protein, or the dystrophin gene, that is. It leads to progressive muscle weakness, eventual loss of function, starting at early childhood, and it afflicts not only the skeletal, but of course the cardiac and the respiratory muscles. The leading cause of death in Duchenne muscular dystrophy is eventual heart failure, but there's a variety of things going on. Typically, the boys lose their ability to walk in their late teens and early 20s, and their death usually occurs in the early 30s. There are very few options available for DMD, which I think I'm going to talk about more in a moment, but just a little bit more on what dystrophin is. Dystrophin, I like to think of it as, and I wrote this in the slide, as as a cushion and of kind of a glue. And without it, the muscles and cells are unable to function properly, and it leads to a variety of different things, progressive damage, and eventually death. Options in DMD are very limited. So the only approved therapeutic right now in this market is Sarepta's drugs, Exondys 51. It only addresses 13% of the DMD patient population. Now, important to note of CAP1002, our therapeutic, we believe we would be complementary to what else is going on in the exon skipping technology companies these days, and we believe that we would meet a need, which of course is the inflammation component of the disease. Most of the boys are on steroids, lifelong regimen of steroids. Steroids, of course, are an anti-inflammatory as well, but they have long-lasting and permanent side effects. So our body of work to date led us to get to the point where we designed our phase 1, phase 2 study known as HOPE in Duchenne muscular dystrophy. And we recently read out results, as I mentioned this morning. We top-lined that data in April. This was a 25-patient study done with 13 boys treated with CAP1002. They received 75 million cells via intracoronary infusion into the heart. And we had 12 patients as usual care. We will have 12-month results available in the next few months. And important notes to take away from this trial are, I believe there are approximately 60 clinical trials ongoing in Duchenne muscular dystrophy. I believe there are 3 or 4 maybe that are only addressing the later stages of the disease, so the more advanced boys in their state. And we were looking at those types of boys. So a majority of them were non-ambulant. They lost their ability to walk. All were receiving steroids. And as I mentioned, a lot of the trial work to date has not been done in this population. This is just a snip of some of the data. More data is available on our website. Our preclinical work, of course, is available on our website as well. And you can see the press release that we put out this morning. But we looked at a variety of different exploratory efficacy endpoints, and the one I want to spend a few minutes discussing is a measurement tool known as the PUL, the PUL, the performance of the upper limb. It was designed specifically for use in the DMD setting out of Dr. Michelle Eagle out of Adam in the UK. And it's a variety of different tasks assessing the shoulder, the elbow, as well as the distal. I think it's 17 or 18 different tasks where boys perform these, according to some of their daily living. That would be very important. And what you can see in some of the, I don't have a laser pointer, but what you can see in the graph on the left is the treated group in red obviously improved at 6 weeks and 3 months. And then you see some of the treatment effect falling off at 6 months. Those are the 3 time points we're looking at. And you see blue, the controls staying near the same. We've shared this data with our thought leaders. Obviously, they found this data extremely exciting. This measurement tool has been highly validated, and we're excited to move forward in our next program in Duchenne muscular dystrophy using this endpoint as our primary efficacy endpoint. We've met with the FDA earlier this summer. They believe that this endpoint can serve as a registration endpoint, which is, of course, very exciting for us. Along with that data was some of the cardiac data. We saw decreases in cardiac scar. You can see again the red in the treatment group, 13 boys that received cells, and blue in control. And then on the right, you see a measurement known as wall thickening, which we also believe to be a principal mechanism of cardiac output. So we saw changes in that as well and improvements in that. So I think you'll see a concordance of the data not only in the skeletal muscle, but also in the cardiac, which is, of course, leading us to the design of our next study. Key conclusions. The results do support the potential benefit of our allogeneic cell therapy to treat boys with Duchenne muscular dystrophy. Saw a signal of skeletal muscle improvement, a signal of cardiac muscle improvement, approximately a 3-month duration effect. We're looking at that now. Our next study is going to be looking at repeat dosing of the cells. So we'll be trying to infuse the cells and then look again a couple months later to treat the boys once again. And CAP1002 was shown to be safe and generally well tolerated. Where we are today is we are in the planning stages, the late planning stages of the next study in Duchenne muscular dystrophy. This could potentially serve as a registration study. We're hoping to initiate it in the first quarter of 2018. This study will be a randomized, double-blind, placebo-controlled study in DMD. One important switch to note is in our HOPE-1 study, we treated the boys using intracoronary infusion in the cath lab, which is a little bit more of a complicated procedure. We've now shifted into IV administration of the cells, which I think is going to be very important, especially when you get into repeat dosing. The primary efficacy endpoint, as I mentioned, will be the performance of the upper limb measured at 6 months. Our principal investigator will be Dr. Craig McDonald. He's out of UC Davis. He's a very prominent figure in the DMD community, and we're obviously extremely excited and delighted that he came on to join our team. The outlook for DMD for Capricor, of course, is moving along the way we had hoped. And we believe, again, we have broad treatment potential. We believe we can be complementary to what's going on in the exon skipping technologies, and we don't have a dependence on specific mutations or deletions with respect to the dystrophin gene. We, of course, could have very favorable pricing for Exondys 51, Sarepta's drug, carries a price tag of approximately $300,000 per year. This is just a snip of some of the leaders, the thought leaders in the DMD community who have joined our scientific advisory board. I put this slide in there because I think there's some figures that I'm sure some people in the room know that are not just focused on the heart. They're across the respiratory function, some neurologists and some people in the community that have really charged forward some of these therapeutics that are moving through clinical development. A quick slide on our manufacturing. Capricor manufactures our cells from donor heart tissue. We have a clean room facility in Cedars-Sinai Medical Center where we're currently making our cells. We have a high yield process in advanced development. And very importantly, we're in late stage selection of a CMO for anticipated early stage commercial manufacture. The finance slide, I always get one slide in the deck, just a snap of our cash. It's healthy cash balance. This was at 6/30 as reported in our 10Q, approximately $12 million. We believe this will take us into June, July of next year, which is not a bad runway rate for us. Capricor's done a really, really good job, at least in my history with the company, of bringing in non-dilutive sources of capital. So CIRM has funded Capricor's 2 clinical trials to date. The NIH has funded some of our preclinical and clinical work. And the Department of Defense is funding some of our exosomes work right now. So we brought in over $30 million in non-dilutive capital, which of course is important from a balance sheet perspective, but it also provides a collaborative relationship with these amazing organizations, and they've really, really propelled us forward. So what to look for Capricor over the next few years or even year? We, most importantly, look for our press release with our 6-month data set presented this morning. We hope to submit an IND for a potential registration study in DMD later this year. We hope to initiate that study in the first quarter of 2018. And along with that submission will go an RMAT designation. And I know they mentioned it in the talk this morning. RMAT is a new designation by FDA, and we're hopefully would add that on. And in our exosome program, we're looking forward to advancing our preclinical work and hopefully filing an IND in HLHS, hypoplastic left heart syndrome, in 2018. So with that, I thank you for having us today. Thank you again to ARM for hosting.